First, to people in the audience, I want to let you know that this is available closed captioned. If you look at that button down in the lower right part of your screen. And also that KK has put a QR code here that will become evident at the end of the talk. And he'd like you to click on that and answer a couple of questions for him to provide a little bit of feedback to him. And I want to start off with a thank you first to KK. He taught me in a little session here how to pronounce his first name, and I'm going to give it a try. But he gives me an easy out because he actually goes by KK. So I should introduce myself too. I'm John Slattery. I'm the Vice Dean for Research and Graduate Education here at the School of Medicine. And Kayode Ojo is our speaker today. Did I come close?
Close enough.
Okay, close enough. And Kayode has, let me tell you a little bit about his background. He got a Bachelor of Science in microbiology from Usmanu Dan Fodio University in Nigeria, which I hope I didn't mangle too badly. And then a professional diploma in laboratory medicine from Obafemi Awolowo University Teaching Hospital in Nigeria, a master's in pharmaceutical microbiology at the University of Ibadan in Nigeria, and then a Ph.D. in pharmaceutical microbiology, also from the University of Ibadan in Nigeria. But he did his thesis work at the Institute for Animal Health in Germany. He then went on to postgraduate training and positions, the first being a teaching assistant in pharmaceutical microbiology and clinical pharmacy at the University of Ibadan. And he was a postdoctoral fellow in pathobiology here. He came across to UW. And also a postdoc in allergy and infectious diseases here at the university. Then in 2009, he became acting instructor in the Division of Allergy and Infectious Disease here. 2012, acting assistant professor. 2013, research assistant professor in AID, and was promoted to research associate professor in AID in 2018. And I should also point out that he's a member and works within the Center for Emerging and Re-emerging Infectious Disease. His title today will be A Case for New Breaks Against Giardiasis Runs. And as you folks know, tuning in, Giardiasis is a GI illness characterized by bouts of greasy, watery diarrhea, stomach cramps, and nausea. It's caused by Giardia lamblia. It's one of the most common waterborne illnesses worldwide, and about 280 people a year become symptomatic with the disease. The treatment relies upon antimicrobial chemotherapy, but about 20% of cases are resistant. And Dr. Ojo then has been using structure-based design to try to overcome that resistance. So structure-based against key enzymes. And then also carrying that work through preclinical novel animal models. So KK, please take it over. Thank you.
Thank you, John, for the introduction, and to the organizer for the opportunity to speak to members of the university community, as well as friends and colleagues from different parts of the globe. Yes, today, as John rightly introduced, I will be talking to you about our research study to find alternative treatment for giardiasis and the tools that we have developed to make it happen. I will start with a short introduction to giardiasis. So giardiasis is caused by infection with the protozoan parasite called Giardia lamblia. Infection can occur by ingestion of environmentally resistant infective cysts passed in stool of infected person or animal. Cysts are really protected by outer shell that allows it to survive outside the body and in the environment for a really long period of time. The amazing thing is that the outbreak is always associated with poor water. One important thing is also that Giardia is virtually never invasive, but it attaches to the lining of the small intestine and through an apparatus called the ventral disc, as you can see in the red arrow indicated here. This ventral disc allows so many of these parasites to colonize a massive area of the small intestine, leading to impairment of the host ability to effectively absorb nutrients, especially fat and carbohydrate from digested food. So Giardiasis is a gastrointestinal disease, and it's characterized by really serious bout of greasy, watery diarrhea, stomach cramp, and nausea. Most of these infections are asymptomatic, and in countries like United States of America or other Western countries, Giardiasis can be likened to a funny kind of disease that occurs most often in travelers, hikers, backpackers, and swimmers in contaminated water. So it's a recreational disease. So the clinical burden in this part of the world is relatively low. Even though the global incidence is estimated to be as high as 280 million symptomatic cases per year. We can therefore say this disease is closely associated with poverty. Impact on children, especially in poor resource country, is particularly profound since this disease can contribute to malnutrition, growth retardation, and diminished neurocognitive development. It's an old disease, been around for a while. And so there are some form of treatment, like 250 milligram metronidazole administered orally 3 times a day for 5 to 7 days. Trinidazole is also quite effective, and there are some other second line of treatment. The big problem is like 1 in 5 of the clinical cases will include the metronidazole resistant strain. Currently, there are no available vaccine. And because most of the available treatment belong to the same chemical series, they are mostly, there's almost always cross resistance across board. So therefore, there is an urgent priority to identify and validate new targets to develop safe, effective, and inexpensive therapeutic alternative. One big problem for all of this, when we think about it, is that reliable tool for drug discovery in Giardia, especially when we think about in vivo mouse model, need to be well developed for us to be able to do this. So the overall goal of my research work is to develop a drug that will cure Giardiasis in mama. So to do this, we have to develop a Giardia strain that can be used in both in vitro and in vivo studies. We want to develop this strain that can be stable and easily readable in a mouse model to test drug efficacy, and also be able to use this model to eventually develop a target-based drug for potential use as alternative treatment. Then we can test this new agent in mouse model. We will also like to define a very good the pharmacokinetic or pharmacodynamic relationship that will be important for in vivo efficacy for after oral use. We are talking about oral use because our goal is to develop a drug that can be used in poorly resourced country, and we don't want them to need an hospital bed or anything before they can take the drug. Resources, yes. We looked around to know what is in there, and we can easily find that there are a lot of experimental methods that are suitable for compound screening. But the issue is still always the mouse or animal model for drug development. And the reason for this is explained in part because of instability in the expression of transgene in Giardia compared with many other eukaryotic cells. The instability leads to variable expression of genes, reported genes in Giardia by some unknown mechanism. The cells, Giardia, can just downregulate any marker gene that you put in or even toss it out by methods that we cannot really fully explain. And we actually found this out ourselves when we engineered a Giardia lamblia strain that expresses a firefly luciferase gene. Every one of us has seen firefly and they glow really well at night. So the gene that makes them glow at night is what we inserted into a Giardia strain under the control of a promoter called beta-tubulin. We really can see that this worked because we can find output of this luciferase expression in Giardia, but it's sufficiently stable for just 24 hours in vitro assay. It lacks a long-term stability over multiple passages to be stable for mouse work. And even after several passages, we completely lost the signal. So we had to go back to the drawing board to ask ourselves, what did we get wrong? Could it be the bad promoter or what? So using an iterative process for refinement of promoter combination against the same firefly luciferase gene, and we targeted an intragenic region adjacent to the trios phosphate isomerase gene locus in Giardia to rule out, is this really a promoter problem? So in these experiments, we use different types of promoter against the firefly luciferase gene. The promoters that we used were the beta-tubulin, the ornithine carbon-mitriphosphatase promoter, the OX promoter, the short and long glutamate dehydrogenase promoter. And our goal was, is it that the promoter messed up our readout, or is it something else? And when you look at this panel, you can see that there is not so much difference in the expression of the same firefly luciferase gene using the different promoter, which tells us like, well, we need to go back and figure it out. So going back, we picked up a widely used promoter, the glutamate dehydrogenase promoter, the long one. This is well used in Giardia and well tolerated. So we know that it can be used very well for both constitutive expression. So this panel shows you what we did with it. We selected different types of luciferase from different sources and then screened them against this promoter. And when we look at this panel, you can see that the expression was different using this long glutamic dehydrogenase promoter against different types of luciferase gene. One important thing is like the click B2 green luciferase was the best expressor, but we also have to think about it. We're dealing with Giardia. A lot of things do happen in Giardia. So we needed to ask ourselves, is this really the best promoter for it, or do we need to do something else? So we did another round of experiments looking at the expression of click B2 green CBG99 luciferase against all the well-used promoters in Giardia. And when you look at this other panel, you can see that the opt promoter was the best one for driving the expression of CBG99 in Giardia. Then establishing that like we can use a lot of different expression is also a question of, are we really can we really rely on it? Because these are parasites that we want to put in mice. We have to think about how fit the parasite are. So we ran a fitness test, and to our surprise, what we found out is like the best promoter for CBG99 was actually the one that showed the least level of signal. But for us, fitness is more important than high level of expression. So we picked this short glutamate dehydrogenase promoter as moving forward to do more experiments. And the next set of experiments was to ask ourselves, under the antibiotic selection that we used to transfer this gene into the parasite, if we remove those antibiotics, are these expressions still going to be there for a long time or not? And then we grew this parasite in the presence and absence of the antibiotic for 4 weeks and found out like there's little difference in this expression over 4 weeks without the presence of antibiotic, which is a huge deal for us because it's kind of confirmed that the robust expression of the green luciferase gene in Giardia can be used for in vivo infection and efficacy studies of potential therapeutic agents. So what we did was now to do that, to test it in mouse model. The first thing to note is that Giardia lamblia is not, mouse is not a natural host of Giardia lamblia. So we had to rely on a technique that will first wipe out the bacterial flora in the parasite. So we using antibiotic, we alter the microbiome. And so that can allow the parasite to colonize, and we can read the presence of the parasite through an improved technique of in vivo imaging system called IVES. This IVES imaging system uses a novel patented optical imaging technology to monitor disease progression or cell trafficking or gene expression pattern in living animal without causing any issue in them. And we can see that with our schedule, we can infect mice and see very robust infection with this new strain of Giardia. We can also not only infect them and see them, we can even develop a model in which we can overlay the growth or the expression of the living cell in a digital mouse so that we can know where these parasites are growing and where they are not. And this is very important because we want to develop a drug that stays in the gut. If we have parasite in one part, we want to be able to see it digitally so that we can improve the output. And so with this, it gives us amazing tool. And we have taken advantage of this new tool to screen almost 33,000 compound library in a phenotypic assay. And this library includes a lot of pharmaceutical library and fungal and natural products. It has actually helped us discover new agents that could be repurposed or further developed as potential new treatment for giardiasis. But for today, I will not be talking about those. This lecture will mostly be focused on our target-based drug development program. Target-based drug development program involve the assay of physiologically important enzymes that are critical to the survival or spread of disease-causing parasite as new potential anti-parasitic molecule for drug development. For our target base, we know that in all cases, these parasites must make essential molecule in form of protein for survival. And translating nucleotide encoded gene sequences into protein is a core process in all biological systems. Since amino tRNA synthesis require enzyme for this process. They are really essential for normal cell functioning. And each tRNA synthase specifically recognizes an amino acid, and they attach it to the corresponding tRNA during peptide chain synthesis. Interference with this process at any point will inhibit the formation of properly charged tRNAs in the ribosome, thus stopping protein translation. For our own case, methionine tRNA synthesis has actually attracted our attention because it plays a key role in translation initiation in most cases, and it is therefore endowed with the unique significance in translational control. But very importantly as well, drug development is very, very expensive starting from the scratch. So we did something that is easier. We did something based on piggyback to an existing program. So because it's cheaper for us. This works very, very well for us because we know that there is a program in existence at the University of Washington that is targeting MET-RS in a research from Dr. Buckner and Dr. Fan's lab that are trying to develop MET-RS inhibitors as therapeutic for human African trypanosomiasis. And this library provided us with an initial 600 compounds that was already synthesized. All we have to do was just to collect data on Giardiasis related by developing experimental data on inhibition of the Giardia enzyme, and then cell assay, as well as pharmacological activity to get insight into what we really need to make a new drug for this or optimize the one they have on hand. So I will give you a little bit of a background on MET-RS. There are two major classes of MET-RS enzyme, the type 1 and the type 2. So human, all of us, we have the two types. The type 1 is the mitochondria enzyme, which is based in the mitochondria. The type 2 is what we have in our cytoplasm. So Giardia lamblia has type 1. Very important to note right here is that the mammalian mitochondria MET-RS enzyme has a compound binding site that is very, very, very similar to Giardia MET-RS. And that is where our major concern for toxicity or side effects of taking our drug will arise, to come up. But we also know that antibiotics like tetracycline that is well used and linosinate have similar issues, and they're still in the market. But whatever the case may be, we really want to develop a drug that will overcome all these issues. One thing is that there is very significant difference with the mammalian cytoplasmic autolog. So for this discussion today, all the compounds that will be discussed will be based on the type 1 enzyme. So before I get into actually describing how we develop the drug, and the best way to start is to imagine how the drug is intended to be used. That is called the target product profile. So we envision having a drug that is an oral 1 to 3 day cure of giardiasis in normal and immunocompromised hosts, whether they have symptomatic or asymptomatic or metronidazole resistant Giardia strain. We also want it to be excellently tolerated and have a lot of good safety profile. And as I said, this is a disease of the poor. We want the cost of the development to be as low as possible. So the good thing is that we started with a lot of data from the TBREW system. So you're looking at here as the molecular basis of how we envision our compound. So example here is a crystallographic complex of some of our inhibitors with the T-Brew enzyme. Very importantly, the enzyme in the T-Brew is very, very similar to the Giardia enzyme. As you can look at the binding site, there are 22 of the 25 amino acid residue in the binding pockets that are very identical to the T-Brew enzyme. So this shows that we can reliably design compounds that will be specific for G. lamblia based on information that we obtain from the T-Brew MET-RS enzyme complex with the inhibitor. Up here are the three compound that represent three of the different compound scaffold that we want to use. There is the linear linker series, the single ring linker series, as you can see, and the double ring linker series. It's really important for us to notice these are just not bringing all the big chemical name, but just visual, like linear ring, single ring, double ring. These compounds are designed to displace methionine from the enzyme pocket by occupying three hydrophobic pockets within the enzyme. You can see as they are in this image. And the very important thing is also that once the compound binds to the methionine pocket, the most amazing thing from the point of drug design is that an auxiliary pocket opens up that is not really available when methionine binds. And then the compound can bind to that auxiliary pocket tightly. This is very unique, and it provides a strong basis for selectivity that reduce side effect to the human host from the treatment. The linker region is where a lot of opportunity to introduce different chemical functional groups actually occur, because the linker region seems to tolerate a lot of changes without affecting the potency of the compound or against the enzyme, since it is just a solvent exposed and it's not binding to any anti-amino acid, but just act as a bridge for the two aromatic warheads. So we tested over 300 T-Brew MET-RS inhibitors from the library against Giardia and MET-RS enzyme and trophozoite. And this table is showing you compound effect on enzyme, G. lamblia cell, human mitochondria, biogenesis, which help us keep, the human mitochondrial biogenesis as a help keep us in line, because for our major concern, which is inhibiting the mitochondrial enzyme activity. So looking at the data, there is SI. A high SI is better than low. You can see from this table, like most of these compounds inhibit the Giardia cells, but in different level. I want to point you to this red cycle. This is where our concerns are. When you see a high SI, that means that the difference in inhibition of the Giardia cell is less than that of the human mitochondrial enzyme. So when we look at the human mammalian cell, liver cell, you can also see that these compounds are not doing anything to, or mostly nothing to, these mammalian cells, which gives us a good feeling that the compounds are not really toxic as much as we want. But I'll go back to the human mitochondrial enzyme. We do this test over 6 days, and it is also in mammalian cells. These cells measure mitochondrial biogenesis And it looks at how the compound affects the protein synthesis in mitochondria. It uses the same FG cell, which tells us that in a short period of time, we may not see any effects by testing the compound. But over a period of 6 days, we can see the effect of the compound on cytochrome oxidase 1 enzyme, COX-1, which is mostly produced in the mitochondria, and it is used as the proxy to see the health of the mitochondria. As I said, that is a major concern developing this series of drugs. So data from these studies guided us to a plan of choosing the double ring linker series as a primary series for optimization, since they actually showed less side effects when we think about it, and the safety and selectivity data of this is better than the other two scaffolds. And when you look at this table as well, you will notice that I highlighted compound 2525 because it has the best profile. So within the double ring linker, we have subseries, and we just want to know which one is the better one. And we are looking at their enzyme activity. Everything look as we expected. The compound inhibit the enzyme, they inhibit the cell, even inhibit metronidazole resistant Giardia cells, and also do not kill mammalian liver cell. The COX-1, which is the measure of toxicity to human mitochondria, we can see a bit of difference, but we like compound 2525. And the reason why we like compound 2525 was also like, at this point, we were not really sure of what pharmacokinetic property is important for in vivo efficacy. So the present compound of choice for treatment of Giardia is metronidazole. It always has a high blood exposure after oral administration. So we decided to go with compound 2525 to test it in vivo efficacy model, taking advantage of what we have, the model we have developed. And reminding you, our goal is to develop a compound that will target enzymes critical to the survival and spread of giardiasis, and providing very, very rapid relief for symptoms of diarrhea or every other symptom associated with giardiasis. So looking at this panel, we normally infect the mice and then wait for 5 days to look at the mice to make sure they have infection. Only infected mice will be recruited for the clinical trial. We look on day 6, and you can see this is day 6. After taking the images, we know that they are all infected. After the end, an hour or so after taking these images, we start treatment. I want you to look at the untreated control and the metronidazole control. I also want to let you know that with those metronidazole at this dosage form, so that it will not cause any issue with the mice. That's one. Then the second is also like, it is not an optimal treatment. Metronidazole treatment also vary from one experiment to the other, just as in the real world. But we look at our compound that we designed, and just a day after the first dose, we can notice that the level of infection went down significantly compared to the control. By day 8, when we gave the second dose, it was done. And then we stopped dosing. And then we started noticing that the infection started coming back. And by day 13, they were all there again. So the question for us is like, yes, this actually worked, but what is it that we did wrong? We had to ask ourselves, did we find a way to make them resistant? But when we think about the dosing period, it does not make sense that resistance would develop in that short period. So the one question we ask ourselves is that this could be that we did not treat enough for a longer time. At this stage, we treated at 50 mg per kg just for 2 days. So we went back to the drawing board and tried it again with a longer dose system. And you can look at this. With the same compound, dose at 40 mg per kg for 5 days. You can see, as a reminder, we dose after day 6, an hour after taking the reading on day 6. And we can see that everything was cleared after day 8, and they did not even come back a week after the last dose. But when we look at the untreated or the metronidazole control, they were still infected. So this data suggests to us that we can clear the infection rapidly, but we may need to keep the treatment longer just to prevent reinfection. So the next series of questions for us is that we really want to develop a drug, but we know that access to newly developed drug in resource-poor regions can be very, very challenging due to a high cost of drug development associated with new drug. So since our goal is to develop a drug that is safe and effective at the minimum possible price of therapy, a strategy to reduce the cost of drug is to find an agent that will not go everywhere in the body, like in the system. And we also think about it like this could be theoretically visible based on the biology of Giardia. I mentioned at the beginning that it doesn't invade the whole cell; it just stays outside and suck all the nutrients from the surface. So if we can figure out a way to design the drug such that the drug does not even get into the blood, but stays within the area of infection, it will reduce toxicity, and we don't have to worry so much about any issues that will be related to the drug in the system. No matter what, when we design drugs that are inhibiting protein synthesis, we always have to think about the issue of bone marrow down the road. The way we're designing this drug is like it should be a short course. Yeah, that should reduce the impact, but you still really want to be on the cautious side. So for this, we study various analog of 2525 that have very similar effect on the Giardia cell, on the COX-1 enzyme, but with very different pharmacokinetic properties. 2525 gets into the blood very well, but we have compounds that barely show up. So we think like if we try those ones, then that may help us reduce the cost of development, but it's a theory, will it work? And so we set out to carry out in vivo efficacy model on all of these different compounds that look like 2525, but don't get into the blood as much as possible. And you can look at this panel as well, starting with compound 2222, going to 2594 and the controls. So remember, we checked all the mice for infection on day 5. After infection, day 6, we check them to make sure before we start treatment, they are still infected, and then we start treatments after day 6. And you can see that by day 7, we see a significant drop in the signal from all the experimental drug-treated mice compared to metronidazole and the untreated mice. By day 8, the infection is more or less done. And you can read that directly in this graph. It's very important to note this graph is a logarithmic graph. So any drop that you see here is over 1,000-fold drop in infection. So it is significant drop compared to the untreated control or the metronidazole treated control. I will remind you again that metronidazole is the drug of choice, but because it has a carcinogenic effect on mice, we almost always want to dose it at a lower level so that we can make sure all the mice are alive by the time we finish the experiment. And we can definitely see in this case that this compound definitely treats the mice and clear the infection. They were dosed at 40 mg per kg, just like 25-25, or even over 4 days. And a week after the last dose, there was no rebounds, except in 2 compounds. The very important thing to note is this: our hypothesis could work, which tells us that we can develop a drug that will treat Giardia that can be cheap and will not go into the blood or cost a lot to develop, because the cost of good will be reduced if we don't have to worry about it going into areas that we cannot talk about or think about, or effect that we cannot predict. So I have highlighted the PK again so that we can see it. These compounds barely get into the blood system. So the effect is localized and they are in the gut, and they can actually perform what we want them to do, which gives us the feeling that we have an hypothesis, and the hypothesis seems to work, that we can develop one drug that would do what we want it to do, be localized in the gut, treats the infection without being expensive and likely having no side effects. So this is where we are in this project. And I just, for today's talk, would like to give you a brief summary of what I've been talking about. So we know we need Giardia new drug. We need new genetic line, new tools to develop the drug. So we developed a new Giardia lamblia cell line that can grow technically like that. And we've confirmed this using different types of acid. We've also used it to screen Giardia lamblia library to find new drugs that can potentially be used for treatment of Giardiasis. We've done pharmacokinetic and efficacy study to show that these compounds can really work in vivo. In conclusion, we can say that MET-RS inhibitors have potent anti-Giardiasis activity, and that some of the compounds completely cleared infection in the murine model. Yeah, so the efficacy is independent of blood exposure, which is good for the development. And the next question is, what do we do next? We're still doing more work to find even better compounds. And we're using the structure of the inhibitor binding mode to evolve more potent and even more selective inhibitors. For the ones that we have found now, we're doing more studies to see whether they will not be susceptible to high frequency of acquired resistance. And then we're also doing on them safety tests on this preclinically to identify whether there are some weird metabolite that can be bad for this compound, and doing other tests like escalating the dose to see where we can get any form of toxicity. So far, so good. All the animals look good. And then we will move forward towards large animal pharmacokinetic and efficacy studies. And then finally, we will be doing some other combination with the drug we've found in our new portfolio, and also the ones that we've already known to be relevant in the clinical and used currently. So this work is a huge work, and I'm just the one presenting it. It's a work done by so many people and support from so many groups. I will start by acknowledging member of my lab, Samantha Michael. She was awesome in helping us with the earliest stage of this work. Lynn, Natasha, they've been like very cooperative and coping with my huge demand, especially and workload. Most of our compound have come from the Fang lab, and they are still supporting us. In the UW group, we have Nora, John, Robert, Fred Buckner, and Wesley Van Voice. They've been excellent mentors that have tutored us and helped us, and we bug them a lot for everything. In University of Washington Biology Lab, we have the Alex Paredes Lab that have also helped us with the development of this model. Kelly was the one that came into our lab and said she wanted to do work on Giardia, and she's also been very, very supportive. Our funding is being from Washington Research Foundation and NIH, and thank you for your attention.
Thank you, Ojo. Very interesting talk. I see one question, and please put your questions in the Q&A. The question here, what's the source of reinfection during the short time dosage? Could that not be an indication of resistance? I think you addressed this a bit.
Yes, it could be resistance, but based on the fact that we can go back and treat longer, that says maybe not, but we will not be able to rule out resistance. Also, we can also think about it like we can kill 99% and one just pop up. So yeah, resistance is there, though we've tried to isolate these cells to see whether they are resistant, but we've not been able to confirm that.
Okay, great. I noticed, the doses were kind of high, even for metronidazole. And I suppose that's why you're going through further structure refinement. Do you know anything about water solubility? And I noticed as you talked about some of the tox studies, you weren't seeing much in the way of toxicology yet. And sometimes that's an indication of it's not being absorbed very well, or water solubility problems or something.
Yeah, so these compounds varies in their solubility profile and depending on the pH as well. So the driver of efficacy in vivo, we cannot really link it directly to solubility because we've looked at that and we noticed like they are really varied.
Okay, great. Thank you. Another question here. Since you're trying to avoid intestinal permeability, um, and of course, often we're trying to increase that, do you do any in vitro screening for passive intestinal permeability? So, for example, in Keiko2 cells?
Yes, those are part of experiment that we do. And the case for Giardia is like, it doesn't even need to get into the intestine. It's always just on the surface. So if we can just go through that area, it may dislodge it from the surface that it's attaching to. And
Yeah.
if it cannot attach, it's tossed out.
Yeah, so I think maybe you answered this, but the question was really, if it penetrated Keiko2 cells, that's bad news because you could get toxicity from that.
Mm-hmm.
So I think they're suggesting that Keiko2 in vitro model to be a screen to make sure that the drug stays in the GI tract and doesn't get into the blood. So have you been using Keiko2 cells that way?
No. Why I think that's also something is like, if we cannot measure the drug in the blood, it has or if we have it will definitely get into the blood to some extent, but not as high as possible.
So you can see the questioner here is Laura Shireman, and you might want to talk with her a little bit more about that. Or Laura could contact KK. I'm sure he'd like to talk with you. If there are no more questions, KK, thank you for a very interesting talk. I really appreciate it. Nicely done.
Thank you very much.
Yes.
And thank you for the opportunity.
Great. Thank you. Bye-bye.